Okay, so we'll have a look at the Hardy-Weinberg equations. The Hardy-Weinberg equation is quite straightforward in its root. We start off with p squared plus 2pq plus q squared equals 1. So this equation, if we were to break down each individual bit, it tells us something. Here we have the proportion of homozygous dominant individuals. So we have homozygous dominant here. Here we have the proportion of heterozygous. And then finally we finish off with the proportion of homozygous recessive individuals. So obviously we're dealing with genotypes here. Okay, so in black we're dealing with genotypes. Obviously, if you take all of the homozygous dominant and all the homozygous recessive and all the heterozygous individuals, they will all equal 1. They will all equal 1. If we now consider as well the phenotypes, p squared, being homozygous dominant, will have the dominant phenotype. Q squared, being heterozygous, will also have the dominant phenotype. Q squared, homozygous recessive, will have the recessive characteristic. And obviously, if you take all of those that have the dominant characteristic, all of those which have the recessive characteristic, you will have the total number of individuals. It's important to remember we're dealing with proportions, not true numbers. Now, if we take one further equation, which is important to remember, and that is p plus q is equal to 1. Now, p is the frequency of the dominant allele. Whereas q is the frequency of the recessive allele. Now, how we use these two equations is best looked at through examples. So in this example, the shell of snails may be unbanded or banded. The absence or presence of the bands controlled by a single gene with 2 alleles. That's good for us because we can only use Hardy-Weinberg equation with those particular characteristics. It has to be a single gene and it has to be 2 alleles, only 2 alleles. So the allele for unbanded B is dominant to the allele banded. So unbanded is dominant. That immediately tells me that capital B is equal to P in our equations, lowercase b is equal to Q. It also tells me that if they are unbanded, that is equal to p squared plus 2pq, because if you remember from the previous drawing that we went through, if they're unbanded, they carry the dominant allele. They can either be our homozygous dominant individuals or our heterozygous individuals. They will all be unbanded. So, the population of snail contains a 51% unbanded. 51% unbanded. So that also tells us that p squared plus 2pq is equal to 0.51. That's how we express a percentage in this equation. So use the Hardy-Weinberg equation to calculate percentage you would expect to be heterozygous for this gene. So we're trying to find these individuals, the 2pq individuals. In order to do so, we've got to use both equations. So if we know that p squared plus 2pq is equal to 0.51, we know as well that 1 is equal to p squared plus 2pq plus q squared. which means that 1 is equal to 0.51 plus q squared. We rearrange it a little bit, that means that q squared is equal to 1 minus 0.51, which means that q squared is equal to 0.49. So then if I square root 0.49, that tells me q being the square root of 0.49 is going to be equal to 0.7. So if we then use the equation we started off with, p plus q equals 1. If q is equal to 0.7, p is equal to 1 minus 0.7, which is equal to 0.3. I'm trying to find the individuals which are heterozygous. They're my 2pq individuals, which means that I'm looking for 2 times 0.7 times 0.3. That gives me the answer of 0.42, which is 42%. Okay, so let's move on to one final question. Some results from an investigation are shown in the table. So it gives us a series of years, the season when the data was collected, and the frequency of the B allele. Now, if the frequency of B, that means that is the recessive allele. Okay, now a part of this question which is missing is that the recessive allele is for red colour, which means the capital B is the dominant for black. So dominant for black is capital B. So use the Hardy-Weinberg expression to estimate percentages of red ladybirds and black ladybirds in the autumn 1933 ladybird population. So we're trying to work out the percentage red, percentage black in autumn of 1933. So the data we're looking for is this line here. So if we're looking at this data, it's told us that B, lowercase b, is equal to 0.7. Okay, lowercase, that must be q, is equal to 0.7, which means using p plus q equals 1, we can then work out that p is equal to 1 minus 0.7, which is 0.3. So now we need to work out p squared plus 2pq plus q squared is equal to 1. P squared is 0.3 squared plus 2 times 0.3 times 0.7 plus 0.7 squared is going to equal 1. Now, in order to answer the question, we need to find the percentage of red ladybirds and the percentage of black. The red ladybirds, as we've already mentioned, red are the recessive. That means that red is going to be 0.7 squared, which is going to be 0.49. Okay, so it's 0.49. That's going to be 49% red ladybirds. Obviously, that must mean that black is going to be 51%, but we could work that out as well. 0.3 squared is going to be 0.09, and 2 times 0.3 times 0.7 is 0.42. Obviously we could add 0.09 to 0.42, that gives us our 0.51. Add that to 0.49, that gives us 1. If we return briefly to the beginning slide, it's important to remember the key to answering any question on the Hardy-Weinberg equation is to think, which data am I being given? Am I being given the characteristic? In which case I'm thinking along the lines that I've just underlined. So if I've got the characteristic, I need to think, is it the dominant, is it a recessive? From there, what can I work out next using the equation? Am I being given the percentage of individuals that have a particular characteristic? If so, from that point I need to be thinking, right, I've been given percentage of individuals which are homozygous dominant. In that case, I'm looking at q squared. Or I've been given the number of individuals that are homozygous recessive. I'm looking at q squared. Where can I go from there? Or am I being given the frequency of an allele, in which case I'm looking solely at this bottom equation? Ask yourself those questions. What information have I been given? What can I do with it? Because using either of the two equations, there is always something that can be worked out from the data you've been given.